Good evening, everyone. So this week we'll start a new lecture series, and the first lecture in the series will be melanogenesis, or the formation of melanin. So melanogenesis is a very essential process. It is as important as keratinization. And to understand the basic disorders of pigmentation, it's very important to understand what is the normal process of pigment formation. How does tyrosine actually gets converted to eumelanin or melanin, and what is the process of color transportation from melanocytes to keratinocytes, so that if we understand the normal process of pigment formation, we can easily understand or at least make some sense of pigmentary disorders. We may also understand fewer technicalities about pigment formation and how different gene mutations lead to different kind of clinical features. Why you have sometimes, collection of symptoms like hearing disorders along with pigment problems, or any neural problems, neural deficits along with pigment problems, or why a theory is given for segmental vitiligo, which includes the migration of melanocytes to the neural networks. So these all concepts will be clear when we know what is the normal process of melanin formation. Melanin, as we know, is the name given to the pigment in the human skin, and it is of two types, eumelanin and pheomelanin. There's also a third small type which is not exactly melanin, that is known as trichrome, but we won't be discussing trichrome much here in this lecture. We'll keep our lectures, we'll first understand the melanocyte physiology, biology, embryology, and also the process of formation of melanosomes and transportation of melanosomes from melanocytes to keratinocytes. Then we will learn about the formation of melanin, starting from the raw material tyrosine, and we go to from tyrosine to eumelanin. And then we'll just see how does UV radiation play a role in pigment formation, or at least increase in the pigment formation. So without much further ado, let's start this week's lecture on melanogenesis. So what are melanocytes? Okay, melanocytes. Melanocytes are specialized cells. They are responsible for synthesis and transfer of melanosomes to keratinocytes. Okay, so melanocytes are the basic cells. So here is a diagram or a picture, an image of melanocyte, a diagrammatic representation of melanocyte. Okay, now melanocytes are specialized cells which are responsible for formation of melanosomes. And melanosomes are cell organelles that actually contain melanin. Okay, so you have melanin which is enclosed in a melanosome, which is present inside a melanocyte. Okay, and this melanosome gets transferred to a keratinocyte. That is the normal process of melanin formation and transport to keratinocytes. So melanosome are organelles that contain the pigment melanin. Now, in all humans, the number of melanocytes are same. So there's no problem, there's no change or a difference in the number of melanocytes in dark skin individual as compared to light skin individual. It's not like a person of African American origin has more melanocytes as compared to Caucasian race. Okay, it's basically that the number of melanocytes per unit area is actually same or similar in different individuals, but the color difference arises due to the amount, quality, distribution, and concentration of melanosomes inside the melanocytes. So the number of melanocytes per unit area is same in all the individuals. But the difference of the color, the difference of the skin color is because there is different activities of the melanin formation enzymes. There's different quality of the melanin formation. There's difference in distribution of melanin to the keratinocytes, and also the concentration inside the keratinocytes and melanocytes. So because of these subtle differences, you have the intense variation in skin color that we usually see around us. Okay, and this has been a very important aspect to understand, that the number of melanocytes are same. It's just their activity is different from different person to person. So here in the melanocyte lineage, you can see that the cell has a lot of dendrites, and using these dendrites, there's transfer of melanosome to inside the keratin. Gradually, as the lecture progresses, you will understand how this actually happens. Let's move forward. So let's discuss a bit about melanocytes, the key player of melanogenesis, the cell responsible to create melanin. So these are melanin synthesizing cells, and they have a dendritic morphology. So you can see here a lot of dendrites. This thin extension of the cell membrane, sorry, cell cytoplasm. Now they are present at the basal layer along with the basal keratinocytes. They are also present at the outer root sheath of the hair and the hair bulb. So these are the places inside the skin where they are present. The other area that they are present are in the retinal pigment epithelium. But being a dermatologist, we'll discuss only the skin locations. So they are present at the basal layer, outer root sheath, and hair bulb. The nuclei is smaller and oval as compared to keratinocytes. So if you're looking at cells in an H&E section, so if you remember, let's say you're looking at a slide in an H&E section, and let me draw it here so it will be easily visible. Okay, so these are all, let's say, basal cell columnar, and you will see melanin deposition inside the basal keratinocytes. Remember, these are not melanocytes, these are keratinocytes. And you will see a umbrella of melanin on top of the nucleus. So if you are looking at clumped melanin in an H&E slide, most likely you're looking at a keratinocytes because the melanin has been transported to keratinocytes. So melanocytes on H&E will not have that much clumped melanin, so they won't be easily visible. So they are usually not seen. They have a clear halo of cytoplasm. So melanocytes are actually clear or not that easily visible. You should be very much trained in analyzing H&E slides. Okay, so you won't see them very clearly. You might not be able to appreciate the dendritic morphology. But remember, if you're seeing clumped keratin inside a cell, you're most likely looking at a basal keratinocytes, not melanocytes. The number is roughly 1/10 of keratinocytes. That means if n is the number of keratinocytes, it is for melanocyte it is n by 10. For every 10 keratinocytes, you have one melanocyte. Melanocytes don't have hemidesmosomes. Remember in the previous week's lecture we were discussing about hemidesmosomes about in for the last 2 months. The melanocytes don't have hemidesmosomes. Okay, so their attachment to the basement membrane is not that strong. In fact, the reason that they stay at the basal layer is because of their attachment to the adjacent basal cells. So a melanocyte would be like staying here with their dendrites coming out. And the reason it is there at the basal layer is because a melanocyte is attached to the surrounding keratinocyte by cadherin attachments. Let me just draw it again. Let's just say that here is a keratinocyte, keratinocyte, and the attachment with the keratinocyte and the dendrites are the cadherin-based attachments. And these attachments are mainly responsible for keeping the melanocytes at the basal layer. As the keratinocyte migrates upwards, not melanocytes, keratinocytes migrate upwards, the melanocytes stay at the basal layer and they keep on imparting melanosomes to growing basal keratinocytes. Okay, I hope it is clear. If not, let me know in the comments. Let's move forward. Now, this is a picture that I've taken from Rooks 10th edition, page 86.3. Okay, 86.3. And I've changed the color so that it is easily visible. You can see a large cell here. This is the cell membrane. So this whole cell is actually a melanocyte. And if you can look closely, these oval granules, circular to oval granules, these are all melanosomes. So these are melanin containing organelles. Okay, melanosomes. And we'll learn about melanosomes subsequently. So let's understand the embryology of melanocytes, how they migrate from neural crest to their final location in the epidermis. Because if we understand the migration of melanocytes from neural crest cells, we'll understand the various disorders associated to melanocyte migration, gene mutations, and maybe the origin of dermal melanocytosis. So all of this is based if we know the basic process how a melanocyte is formed. Initially, it is known as melanoblast, and gradually it differentiates to form melanocytes. Okay, so melanocytes come from neural crest cells, and neural crest cells are present at the margin of neural tube. So I'll show you a diagram on the next slide, and it will be easier for you. Just remember that neural crest cells are the origin of melanocytes. These are the origin, and this could be a good MCQ question or a Viva question. Okay, question. Now, neural crest cells also give rise to, apart from melanocytes, craniofacial cartilage, bone, secretory endocrine cells, smooth muscles of the heart, vessels, peripheral sensory and autonomic neurons, enteric neurons, and peripheral glia. So now do you understand that if any problem happens at the neural crest level, you will have pigmentary disorders, and you will also have disorders of the neuronal lineage, because the same source is there for both of them. The Schwann cells are also derived from neural crest cells. The melanocytes are also derived from neural crest cells. So if there's a problem at the level of neural crest, you will have problems into two lineages, pigmentation and neuronal problems. That is why a lot of pigmentary disorders like Griscelli's uh, your or oculocutaneous albinisms, all of these are because they have both of the problems. They have pigmentary problems and neuronal problems also. Now from the neural crest cells, so let's just make a rough diagram. This is the neural tube which is invaginating, and eventually it will close. Okay, eventually, but before closure, you will have a certain area at the tip of the limbs, and these are the neural crest cells. From the neural crest cells, here you have just lateral to the neural tube, you have the somite, which gives rise to dermomyotome, which will eventually give rise to dermis and muscles of the body. So what actually happens is you have migration of melanocytes from above, which is known as the dorsolateral pathway, and you have migration of the Schwann cells, the neural lineage, from below, and this is known as ventromedial pathway. Okay, we'll discuss about both of these pathways on the next slide. Just remember that the dorsolateral pathway is the major pathway for migration of melanoblasts. Okay, and some of the melanocytes or some of the melanoblasts also migrate from the ventromedial pathway. But the major pathway for migration of melanoblast is the dorsolateral pathway. The ventromedial pathway is responsible for Schwann cell migration, and some Schwann cells also form melanocytes. Remember, they have the common lineage, they have common ancestors. So they can differentiate two melanocytes, and some melanocytes are derived from ventromedial pathway, while the major pathway for migration of melanoblast are dorsolateral pathway. Let's move forward. At roughly about 50 days, half of the melanoblasts actually reach hair follicles and epidermis. So while the melanoblasts are migrating from neural crest cells and eventually reaching the skin and hair follicle bulbs, about half of them roughly about takes about 50 days to reach there and stay there. And during that migration, gradually they are converted from melanoblasts to melanocytes. At roughly about 120 days, they become pigmented, and then they will be pigmented throughout the life. Okay. The migration also takes place into additional sites like uveal tract of the eye, which includes choroid, ciliary body, and iris, the leptomeninges, and cochlea. Okay, so that is why when there's a problem in migration of melanocytes, you can have hearing issues because cochlea is also there. Okay, it is said that the melanoblasts are responsible for proper development of inner hair cells. So if the melanoblasts are not able to reach cochlea, the inner hair cells won't be formed and there will be hearing deafness, sensorineural deafness. They're also there in the leptomeninges. So that is why you have neuronal issues in case of, let's say, pigmentary issues in oculocutaneous albinism, you can have neuronal issues because of same reason. The eye involvement is there because remember, apart from skin and hair, the other parts where melanin are deposited is the retinal pigment epithelium. And retinal pigment epithelium is in adults. That means when the child is born, the additional sites, there is absence of melanocytes, there's absence of melanoblasts, because during the gestational development, slowly these migrated melanocytes will die off and they won't be there. So in adult body, the melanocytes are eventually in skin, hair, maybe a bit of skin, hair, and retinal pigment epithelium. So these are the sites of melanocytes in adults. The migration are controlled by many factors. There are many factors that control migration, but the most important or the major factor is KIT-KIT ligand interaction and endothelin receptor type B. These are responsible for proper migration of melanoblast to the ventral site in the skin, and we'll learn about each of them specifically in this lecture. So let me reiterate the formation. Okay, so this is the notochord. Sorry, this NO is the notochord, and this NT is the neuronal tube. When the neuronal tube is invaginating like this, so at the level of crest, you have the neural crest cells. So here you will have neural crest cells. Gradually, the cells will migrate, and this is the dorsolateral pathway, while this is the ventromedial pathway. Okay, the dorsolateral pathway is responsible for the formation of the epidermis, which is derived from ectoderm, and the ectoderm will have melanocytes from the dorsolateral pathway, which is the major pathway. So dorsolateral is the major one. There is minor migration from the ventral pathway, but remember that the ventromedial pathway is important for neuronal migration. Okay, so this ventromedial will make Schwann cell precursors, nerves and everything, while the dorsolateral will form melanocytes. So here you can see the neural tube, the neural crest cells, and then dorsolateral pathway migration, and this is ventromedial pathway migration. Clear? Let's move forward. So here in mammals, the cell at the neural crest cell is actually a sort of stem cell, and this can modify into two different lineages. You have stem cell, and this can either be a melanocytic lineage or it could be a neuronal lineage. Okay, so what happens is you have neural crest cells which modifies into bipotent melanoblast glia progenitor. So you have melanocytic lineage and you have neuronal lineage. Okay, under the action of SOX10 plus gene, this gene, it gets transformed into melanoblast. And in melanoblast, with the interaction from the KIT ligand, DCT, and MITF, we'll discuss about MITF in subsequently. This converts into melanocyte stem cell, and this melanocyte stem cell is there at the level of hair follicle. So from hair to the hair follicle, you are having the dorsolateral migration of the cells. Clear? And then these acts as a source, and from follicle you go to the intrafollicular epidermis. So this is a part of skin. These are the follicles. The pigments here are known as follicular melanocytes, and in between them you have the interfollicular epidermis, and in interfollicular epidermis you have interfollicular melanocytes. Remember, these are rich in stem cells. The follicular melanocytes are rich in melanocytic stem cells. So in vitiligo, you see perifollicular repigmentation. Why is that? Because the stem cells are present at the hair bulb and outer root sheath cells. So under stimulation by treatment, they start producing pigmentation. Okay, let's move forward. Okay, so after looking at the genes and transcription factors, let's discuss the hero of melanogenesis, the melanosomes, or the organelles which actually stores melanin. Okay. Now, melanosomes are round to oval. They are membrane-bound organelles, and they are present in melanocytes and retinal pigmented epithelium in adults. Clear? Now, arbitrarily, the formation of melanosomes is divided into four stages for convenience purposes. So, four stages are there. So you can see here stage 1, stage 2, stage 3, stage 4, and we'll learn what are the changes as the melanosome goes from stage 1 to stage 4. Few melanosomes, or the melanosomes that has few melanin. Remember, there are two kinds of melanin. You have eumelanin and you have pheomelanin, or basically black melanin and red melanin. Okay. So few melanosomes are those melanosomes that have few melanin instead of eumelanin. They are round, smaller, disorganized, and has less dense fibrillar matrix. Now, what do we mean by that? Can you see this wires like structure in stage 2? This is the fibrillar network, and this is responsible for migration, transport, uh, collection, storage of melanin inside the melanosomes. These wires are responsible to collect melanin inside the melanosomes. Okay, just remember that. A very important aspect of this fibrillar matrix network is PMEL amyloid protein, and it is very essential to manage melanin and melanosomes. It acts as a scaffolding, it acts as a framework, and on this framework, the melanin pigment is deposited inside the melanosomes. In other words, you have an oval melanosomes, okay? You have the fibrillar network here. It is arranged parallelly over the long axis, and over this axis, you have deposition of melanin. And this PMEL protein is responsible for deposition and collection of melanin inside melanosomes, and that makes it very important. This could be very well a good quiz question. Let's move forward. So in this diagram, you can see stage 1, not so much melanin, sorry, not much melanized. In stage 1, the cell is round, roughly zero point 0.3 micrometer. There is no tyrosinase activity. Remember, tyrosinase is the major enzyme in melanogenesis, so that is not working. It is small and it has intraluminal vesicles. So you can see intraluminal vesicles here. Okay? So this whole is the big cell, stage 1 melanosome. It is not melanized. When we go to stage 2, it becomes oval. Can you see the oval shape now? Look at this oval shape. Okay, oval shape. The tyrosinase activity is high. There is parallel fibrillar matrix. Can you appreciate these wires here? Okay, here inside the cell, they are running longitudinally like this. So this is the parallel fibrillar matrix. This cell is also not melanized. The melanin synthesis begins here. This could be a good Viva or MCQ question. That melanin synthesis begins at stage 2. In stage 3, the tyrosinase activity is high and the melanosome is now melanized. So can you see this black color? This is melanin. And in stage 4, the tyrosinase activity is now decreased to a minimum and the melanosome is fully melanized. So much so that you are not able to fully appreciate the parallel structures. So here you can see only melanin. The parallel structure is not able to see properly because it is hidden by melanin. So now you understand the various stages of melanosome formations. Let's move forward. Now, melanocyte density. Remember that the density is similar in humans, different humans, and the variation in skin color is mainly because of the amount of melanin, the process of melanin, the oxidation reduction of melanin, okay, and the distribution of melanin to keratinocytes. It all depends on the process. It's a physiological process, while the number of melanin density or the number of melanin per unit area is essentially similar. It is seen highest at the level of highest at the level of genitals, roughly about 1500 melanocytes per millimeter square. Millimeter square is roughly like this area, this small area. And the face, these are the maximum area in which the density is very high, while minimum is seen at the level of trunk, roughly about 900 per millimeter square, and also extremities. Chronic sun-exposed skin can have 2 times the number of melanocytes and increase in size and activity. So if in one individual, let's say here is an arm, okay, here is an arm and the palm is facing you. So if this is the palm, here is the exposed area and here it is not exposed area. So the exposed area will have 2 times melanocytes as compared to the not exposed area in the same individual. Remember, the density is same in different individuals. There's a concept known as epidermal melanin unit, which was given by Fitzpatrick. Okay, so what essentially is there is you have a dendritic You have a dendritic melanocyte. Okay. And then you have different keratinocytes attached to a single melanocyte. Different keratinocytes. So this is keratinocyte. Epidermal melanin unit is the connection of one melanocyte with its all connected keratinocyte. So essentially this whole is known as epidermal melanin unit. So Fitz and Rooks mentioned one melanocyte with 40 keratinocytes roughly, while Bolognia mentions one keratinocyte to 30 to 40 keratinocytes. Okay. So I just mentioned both of them. But remember, epidermal melanin unit is the collection of one melanocytes and attached keratinocytes, which is about 1 is to 40 or 1 is to 30 by 40 if you remember Bolognia. So one melanocyte attached keratinocyte is epidermal melanin unit. Let's move forward. So let's discuss about melanin, and after discussing about melanin, we'll discuss about the melanin synthesis, the pathway. And the pathway is people find it very difficult to remember. I had difficulty in remembering during my JR ship. I remembered it during my SR ship. And we'll also discuss a unique mnemonic that I used to take help whenever I needed to remember the steps of melanin synthesis. Let's start with melanin. We have two types of melanin, eumelanin, pheomelanin. Okay, eumelanin is brown black, but pheomelanin is yellow orange or red melanin. Okay, eumelanin shields against UV radiation. That means it is UV protective. It scavenges UV induced reactive oxygen species. So all the UV induced damage, eumelanin is the melanin that protects. So the darker skin, the more brown skin you have, the more black skin you have, you are protected against UV. And we should be thankful of our Indian complexion. We should not run after fair complexion, because remember, UV damage is a very serious thing. Okay, and fair complexion is temporary, UV damage is permanent. So make sure. Pheomelanin may be a pro-oxidant. That means it will increase reactive oxygen species. And because of that, it will increase UV damage. So UV damage is causing reactive oxygen species formation. Pheomelanin also causing reactive oxygen species. There will be further DNA damage. That means pheomelanin is not good. And that is why people with red hair, or redheads as they are known in Western Hemisphere, they are more prone to UV-induced damage and melanomas. Also, freckles and lentigens are more common in them, the fair-skinned people. So, we should be proud of our brown and black skin. Let's move forward. Okay, so let's start the process of melanin formation. Just take about 30 seconds and look at the whole process. Don't remember anything. Just look at the process and try to internalize what actually is happening. So we have started with tyrosine and gradually will form all the intermediates. Here it divides into two parts. Okay. If cysteine is present, okay, if cysteine is present, that means sulfur containing amino acids are present. You will have a sulfur containing molecule, and this sulfur containing lineage will lead to pheomelanin. Remember, if cysteine is not present, then non-enzymatically L-dopaquinone gets converted to leukodopachrome. Okay, look here, just look here. Here you have only one cycle, and this molecule is free. While in leukodopachrome, you have two cycles. There is cyclization of the molecule, and that is why leukodopachrome is also known as cyclodopa. Remember this name, cyclodopa. After cyclodopa is formed, you get dopachrome, and dopachrome means that melanin will be formed. This is the essential molecule in melanogenesis. Dopachrome gets divided into two parts, and both pathways eventually leads to eumelanin. Okay, one has mainly quinone, while other has carboxylic acid along with quinone. So here you have the COOH group along with a ketone group. And here you have only ketone group. Okay. So just remember this. Can you see this ketone groups here? Quinone groups, that's all. So this is the overview of melanogenesis. Now we'll look at each and individual steps and understand the process of melanogenesis. Okay, so be ready. Let's move forward. Now this is the first step. You start with the amino acid tyrosine, and we get tyrosine from phenylalanine. Phenylalanine. And phenylalanine or L-phenylalanine is converted by the action of L-phenylalanine hydroxygenase. Hydroxygenase. And this leads to formation of tyrosine. Now tyrosine under the action of tyrosinase enzyme forms L-dopa. So L-phenylalanine leads to L-tyroxine leads to L-dopa. Tyrosinase related protein 1 or TRP1 stabilizes tyrosinase, which is the main enzyme of melanogenesis. This step is very crucial. Tyrosinase is inhibited by hydroxyquinone. Okay, it is inhibited, and that is why you use hydroquinone in treatment of melasma and pigmentary disorders. Clear? Tyrosinase is a copper-containing enzyme. Copper-containing enzyme. So in disorders like Menkes disease, in which copper transfer is not proper, you will have a bad functioning tyrosinase, and that is why you see pigmentary dilution of hair in Menkes disease. Okay, now you got everything. The first step is L-tyrosine to L-dopa via the enzyme tyrosinase. Let's move forward. From L-dopa, again by the action of tyrosinase, you form L-dopa quinone. Quinone. So look at the quinone group. Here are the quinone group. Okay, L-dopa quinone. Just remember the word quinone. Let's move forward. Now L-dopa quinone, if there is cysteine present, will go and form pheomelanin. If there is no cysteine, it will automatically get shifted to leukodopachrome, which is also known as cyclodopa. Now can you see there is no cycle here, but here you can see two cycles. So there's cyclization of the molecule. You have cyclodopa. So L-dopa becomes cyclodopa. And also look here, the quinone groups are removed. They are not here now. They are here. They are present here, but now they are not there. So the major part which has happened is cyclization and loss of quinone. So the quinone is gone and you have cyclodopa. You have cyclodopa. Clear? Cyclodopa will eventually form eumelanin. Let's move forward. Now cyclodopa gets converted to dopachrome, and dopachrome is an essential molecule. Remember, look here. The OH group is converted to a ketone group. So you have dopachrome. Chrome is color. Okay, dopa is the molecule. Leuco means white. So leukodopachrome doesn't have color. It oxidizes. Oxidation of alcohol causes forms ketone group. Okay, it oxidizes to dopachrome spontaneously. You don't require any enzyme spontaneously. And formation of dopachrome means now eumelanin will definitely form because dopachrome is colored. Now you will have pigmentation because of this molecule. Let's move forward. Now dopachrome, there is removal of this COOH group and you get oxyindole, dihydroxy, two groups of hydroxy, so dihydroxyindole. So dopachrome converts to dihydroxyindole. Let's move forward. Now dihydroxyindole, again the action is the same enzyme tyrosinase. So now you remember how important this enzyme is and why mutations in the tyrosinase enzyme has a lot of problems in pigmentation. It's because of this. So dihydroxyindole, that means at the carbon 5 and 6, you have two hydroxy groups. But here you can see the hydroxy groups are changed to quinone. So you have 5,6 quinone. And you have an indole ring. This is an indole ring. This ring here is an indole ring. So 5,6 dihydroxyindole under the action of tyrosinase changes to indole 5,6 quinone. Clear? Let's move forward. And this indole 5,6 quinone then gets converted or dimerizes by itself and forms eomelanin. And this eomelanin is formed by dihydroxyindole. That is why it is known as DHI melanin. It is black, insoluble, and has high molecular weight. Okay, eomelanin. But remember, I told you that dopachrome is shuffled to two pathways. We followed one pathway to form eumelanin. We should also see what the second pathway forms. The second pathway also forms eumelanin. It just takes part in a further step of reaction by adding a carboxylic acid group. So let's see that path. So let's go back to dopachrome. And dopachrome under the action of tyrosine related protein 2. Okay, tyrosine related protein 1 was the one at the first step stabilizing tyrosinase. Tyrosine related protein 2 also known as dopachrome tautomerase. Tautomerization is a change in structure. So tyrosine related protein 2, what it does is it adds a hydroxyl group to this ketone group, and now it is 5,6 dihydroxyindole 2 carboxylic acid. Here, 2 carboxylic acid. Remember, in the first step, the carboxylic acid group was removed. Here, it is not removed and it stays to form dihydroxyindole. This is known as DICA, dihydroxyindole carboxylic acid, DICA. Let's move forward. Now, DICA under the action of tyrosine related protein 1. Again, from step 1, forms indole 5,6 quinone carboxylic acid. So it's very easy to remember these names. I know it may sound overwhelming, but when we'll see the larger picture together, it will be easier to remember. Plus, I will give you a mnemonic to remember everything. Let's move forward. Just remember that this carboxylic group is there. If the carboxylic group is not there, you have the simple indole 5,6-quinone, which we see in the first step. But now we have just an additional addition of carboxylic acid. And this is what forms again eumelanin. But since it is a carboxylic acid mediated, you have DICA, dihydroxyindole carboxylic acid melanin. It is brown, slightly soluble, and has intermediate molecular weight, and that is why eumelanin is known as brown black melanin. Brown black melanin. It is DHI melanin. Brown is DHICA. Now look at the whole process and you'll be easily able to understand. Tyrosine to L-dopa, L-dopa to L-dopa-quinone, L-dopa-quinone to cyclodopa, cyclodopa to dopachrome, dopachrome to dihydroxyindole, dihydroxyindole to indole 5,6-quinone, 5,6-quinone to eomelanin, dopachrome to dihydroxyindole carboxylic acid, indole 5,6-quinone carboxylic acid, eomelanin. That is all you have to remember. You may not remember fumelin, that's okay. In exams, you can just write cysteine glutathione and directly to fumelanin. But you should remember the steps of formation of eumelanin because that will be asked in the exam and you have to draw the flowchart. No, do not draw the structures. Please do not draw the structures. Just keep writing the name of the chemicals. So let's understand this mnemonic for melanogenesis, which I was using. Those leaving quality classes don't develop in education. Okay, those who don't take quality classes, don't know proper classes, they won't have good education. So remember this sentence: those leaving quality classes don't develop in education. Let's see the mnemonic. Those means tyrosine, leaving means L-dopa, quality means L-dopa quinone, cyclodopa, dopachrome, dihydroxyindole, indole, eumelanin. So these are the structures in the formation of melanin from tyrosine to eumelanin. Those living quality classes don't develop in education. T for tyrosine to L-dopa, L-dopa to L-dopaquinone, L-dopaquinone to cyclodopa, cyclodopa to dopachrome, dopachrome to dihydroxyindole, dihydroxyindole to indole 5,6-quinone, and eumelanin. So using this mnemonic, you'll be easily able to remember the steps. Just remember that tyrosinase acts here, tyrosinase acts here, and tyrosinase also acts here. Clear? Now, the other pathway to form eumelanin is dopachrome, dihydroxyindole carboxylic acid, indole 5,6-quinone carboxylic acid, eumelanin. So use this mnemonic, practice this mnemonic, and you will never forget the process of melanogenesis. Let's move forward. Now the melanin has formed. We have deposited the melanin inside the melanosomes. Now the melanosomes are ready to get transferred to keratinocytes. Stage 4 melanosomes get transported to dendritic tip, and this transportation is mediated by kinesin. So let's make a keratinocyte here. So it has a lot of dendrites, okay, and melanosomes denoted by blue are formed in the cell body, and these are stage 4 because they are melanized. They will gradually move towards the tips and they will get accumulated at the tips, and from the tips they will go inside keratinocytes. Okay, so this is how they get transferred. The melanosomes get transferred from melanocyte to keratinocyte. The melanosomes are captured at the tip and accumulated. This is the tip of melanocytes, and this capture is done by RAP27A melanophilin actin complex. You need not remember, this is just extra information. Mutation in this complex leads to Griscelli syndrome, partial albinism with immunodeficiency. Because while immunodeficiency, remember melanosomes are derived from lysosomes because they are membrane bound organelles that has all the tyrosinases and all the aces enzymatic activity. So the lineage is same. So if by any chance there is a problem in the transportation of lysosome and lysosome related vesicles, the melanosomes won't be get transferred. So there will be partial albinism and the lysosomes will not be transferred. There is immunodeficiency. Understood? Now you're getting the concept why Griscelli syndrome has immunodeficiency along with pigmentary dilution is because of this. Okay, so now we know the complex at the level of melanocyte. At the level of keratinocyte, you have keratinocyte growth factor and protease activity receptor 2, PAR2, which is at the level of keratinocyte to take the melanosomes inside. So the RAP27A melanophilin actin complex or the Myo 5A complex gives the melanosome, and the keratinocyte growth factor and protease active receptor takes the melanosome inside the keratinocytes. And now the melanosomes are inside the keratinocytes. So this is a diagrammatic representation of the same process that I said. It is taken from Bolognia. So here the stage 4 melanosomes, this is a stage 4 melanosome, gets transported from the center of the cell to the tip where it is captured by the Myo 5A complex and it is kept there. Under the action of UV radiation, this transportation increases, and that is why more melanosomes are transferred to the keratinocyte, leading to UV-induced pigmentation. Okay, UV-induced pigmentation. So this is the myo-5A protein, and these are the actin filament. This transportation this is known as anterograde migration of melanosomes, while this one is known as retrograde because the melanosomes are coming back. And this retrograde is mediated by the different microtubules, while the anterograde is by dynein and kinesin. Let's move forward. So there are at least 4 theories or 4 processes by which melanosomes get transferred into keratinocyte. First is zygophagocytosis. Cytophagocytosis, that means cell will eat a part of cell. So here, there is the melanocyte and here is the keratinocyte. It will eat a part of the melanocyte, and here that is how melanosomes are transferred into keratinocyte. Clear? That is known as cytophagocytosis. Membrane fusion. In membrane fusion, the membranes of melanocyte and keratinocyte fuse together to form a tube. And then this tube is used as a pathway to give melanosomes to keratinocyte. Let's move forward. Shedding phagocytosis. In shedding phagocytosis, there is shedding, there's breakage of this area, and it melanosomes are free for some time, and then this free is taken up by keratinocytes, and then you have keratinocytes. So this is the third way how melanosomes are transported to keratinocytes. And exocytosis, endocytosis. That means you release the melanosome. It is not bound by organelle. The difference is not bound by a part of cell membrane in this process. And then it is taken up by a keratinocyte. So you leave the melanosome there and the keratinocyte takes it. There's no major pathway. All of the four processes have been shown to work. And that is why we cannot say what is the major pathway in humans or not, but all the four processes have been proven to work. That is why it's important to mention. In exams, you don't have to draw these diagrams. You just have to mention that using four pathways, melanosomes are transported to keratinocytes. The four pathways are cytophagocytosis, membrane fusion tube, simple exocytosis, and exocytosis, endocytosis. Okay, let's move forward. So here you can see an epidermal melanin unit. This is the melanocyte, and it is attached to various adjacent keratinocytes, and it is slowly transporting these melanosomes inside those keratinocytes. We are clear? Now we have learned about melanocyte. We have learned about melanosomes. We have learned about the formation of melanin. We have learned about the transportation of melanin inside the keratinocyte. Let's move forward. So now the melanocytes are there inside the keratinocyte. Okay. Their uptake, they are taken up by the keratinocyte and then using dynein mediated retrograde transport. Retrograde means from periphery towards nucleus, they reach the nucleus. Okay. So if this is the cell and this is the nucleus, this is known as anterograde, and this is known as retrograde, backwards. So there is retrograde transport, which is mediated by dynein. All the melanosomes are collected on the top of nucleus, and that is known as capping of the nucleus. In other words, like a cap, they cover the nucleus. Okay. So here we can see a basal cell. This is a basal keratinocyte. Basal keratinocyte. This is the nucleus. And here you have the here you have the transported melanosomes taken inside the keratinocytes. Okay. And they will get collected and shifted retrogradively over the nucleus, and this will form a melanosome cap. A melanosome cap. And this cap is there to protect from UV radiation because this is where the UV light is coming. Okay. UV light is coming from this direction. So it acts like an umbrella, a cap to save the nucleus. Why the nucleus? Because DNA is present here. So you need to protect the genetic material, and that is how melanosomes form a cap to protect the DNA material. So in light skin, the melanosomes are present in clusters of 4 or 8 or small, while in dark skin, they are large, individually present, and numerous, multiple. We're talking about melanosomes. As the basal keratinocyte migrates from basal cell to the stratum corneum by the process of keratinization, this cap is also eventually lost. So when a basal cell migrates to form the corneal cell, the stratum corneum cell, the corneocytes are thin and all the organelles are more or less lost. And this being an organelle at that point of time is also lost as the cell moves up. So it is degraded by autophagy. Autophagy means the cell itself degrades the organelle. So this is how the journey of melanin, which starts from tyrosine, gets converted to eumelanin, gets incorporated in melanosome, gets transferred to keratinocytes, forms a nuclear cap, degraded by autophagy, and the whole life cycle of melanin completes. Now, this special section is only for the members, and we'll discuss very quickly by the MSH-MC1 receptor or MITF pathway. Now, what is MC1R? MC1R is melanocortin 1 receptor. There are multiple receptors. I think there are 4 or 5 receptors, and MC1R is the major receptor. It's a G protein coupled receptor. It determines eumelanin or pheomelanin. What melanin will be formed inside the cell is regulated by MC1R. If the MC1R is inactivated, it causes red hair phenotype. Red hair phenotype means fair skin, red hair, and inability to tan. And if there is inability to tan, there will be increased burning of skin. So if there's no tanning, there will be burning. Tanning is a protective measure of skin. So there will be burning. Clear? This pathway, the MC1 receptor activation, is controlled by MSH or melanocyte stimulating hormone. And from where do we get MSH? We get it from POMC or pro-opio melanocortin. So POMC gives rise to alpha MSH, which is the main activator of MC1 receptor. Okay. Now, from the pro-opio melanocortin, alpha MSH is not the only thing. From pro-opio melanocortin, you have ACTH, you get alpha MSH, and you get beta endorphins. You also get beta MSH, you also get gamma MSH, but alpha MSH is the main stimulator of melanocyte. This is just a reminder of how a G protein coupled receptor works. G alpha S dissociates from the receptor. It increases cyclic AMP, which activates protein kinase A, which activates protein kinase CREB, which leads to DNA response element. And in DNA, it increases the activation of MITF. MITF is the main gene of melanogenesis. Main gene. Main gene. So, and this is how MSH leads to formation of melanin by activating MITF. Phosphodiesterase 4 is a negative feedback for MITF generation. That means increasing phosphodiesterase 4 will decrease MITF to act as a check and balance to this activation of MITF. Let's move forward. UV induced tanning. I think we are just ending the lecture soon. So UV induced tanning is two parts, early and late. The early tanning occurs in minutes to hours. It's basically because of UV exposure, mostly seen in dark-skinned individual, and it is because of oxidation and redistribution of melanosomes. So what happens is within minutes to hours of UV exposure, the melanin gets oxidized and redistributed and leads to a short-term tanning, which gets removed in few minutes or let's say a few hours after the UV exposure is stopped. Since it is temporary, because it happens quickly and goes away quickly, it is temporary. It is not UV protective. UV protection occurs from prolonged exposure to UV. This leads to late tanning, which takes about 3 days to about a week to develop. It is mostly because of UVB and shorter UVA to some extent. And the main process is increased melanocyte density and transfer. Okay. So in the early tanning, the color was because of oxidation and redistribution of melanosomes. In late tanning, it is because of increased transfer of melanosomes to the keratinocytes. There is also mild increase in number, but the major process is increased melanocyte density and transfer. So now you remember why processes like stupid detan doesn't work. Tell your patient it doesn't work, it causes problems. As a doctor, we must tell people not to go into these kind of shenanigans. Processes like detan, scrubbing, useless. Know the process of melanization. You'll be able to counsel your patients much, much, much better. As we have already mentioned, that density of melanosomes are roughly 2 times as compared to non-exposed region. Okay, in the exposed UV exposed region, you have twice the number of melanosomes and melanocytes. Let's move forward. Now, these are the points through which UV radiation increases tanning. So UV radiation increases POMC transcription. So more PMs, POMC, more alpha MSH. TGF beta decreases MITF and UV radiation decreases TGF beta. So MITF is inhibited by TG transforming growth factor beta 1 and UV radiation decreases TGF beta. So eventually what is happening? It is increasing MITF. Negative, negative, na? Dost ka dushman dost. Same thing, okay? So UVR increases POMC, UVR decreases TGF beta, which is the inhibitor MITF. UVR increases cytokines growth factors by keratinocyte. It increases the concentration of mineralocorticoid 1 receptor. It also activates protein kinase C. Remember in the G protein coupled pathway, it's an important molecule. It increases nitric oxide production, which increases cyclic GMP pathway. Again, the same reason why more MITA will be formed. It increases the activity of tyrosinase. Tyrosinase also induces formation of MITF. It's a self-positive feedback loop. From all these points, there is increase in MITF, and we all know by now that if MITF increases, melanogenesis increases. This is how more melanin is formed. UV radiation also increases melanocyte density and transfer of melanosomes to keratinocyte, and this is majorly responsible for the late type of UV tanning. So all these points that I mentioned here is responsible for UV induced tanning. The process why UV leads to increased pigmentation is because of these. Frequent new research is coming up. We will have multiple points in the future, but this is how UV actually darkens the people, darkens individuals. So this diagram I've taken from Fitz, page 354, sorry, 344. And here we can see UV light causing DNA damage, and this DNA damage activates P53. P53 activation actually increases POMC. POMC increases alpha MSH here. Alpha MSH acting on MC1 receptor causes MITF. MITF leads to increased melanogenesis, increased melanin formation. This melanin then gets transferred vapis into the keratinocyte, and this here it will form a nuclear cap and protect from UV radiation. So now you get the cycle. UV light to DNA damage to P53, P53 to DNA to POMC, POMC to alpha MSH, alpha MSH to melanocortin 1 receptor, melanocortin 1 receptor to MITF, MITF to melanin, melanin to melanosome, melanosome back to keratinocyte. Inside the keratinocyte, nuclear cap protects from UV light. So now you understand why UV exposure actually leads to formation of melanocap and protection from UV light. Now you're getting the concept clearly. Let's move forward. So with that, we finish this humongous lecture on melanogenesis. Just remember, I've covered everything: melanogenesis, melanin, transportation of melanin, and melanoma risk, UV-induced tanning, which is a very important process to know at the residency level. Further, if you want to know, you can read chapter 20 of Fitzpatrick, 86 of Rooks 10th edition, 65 of Bolognia's Dermatology. But remember, all 3 books are condensed into this one video. Go through this video again and again. You'll be easily able to understand the complex process of melanogenesis. These 2 articles are very good if you want to go in depth about genetic control of melanogenesis. They are very detailed, very good articles if you want to go. But I've taken whatever is important from them and put this into the video format. So with that, we'll cover. It's a very long, more than an hour long. At least it covers all the basic concept of melanin formation. Okay, and it makes it easier. I hope people go through it and you make use of the mnemonic that I've given. Okay, those living quality classes don't develop in education. Remember this line. You will remember all the steps of melanin formation. So I will not take further your time. It was a long lecture. I did it one take, so I'm also tired. I have to go and have dinner. Thank you very much. Any doubt, suggestions, any queries can easily be put in the comment section so that I can reply to them properly. Or you can email me directly, but I prefer the comment section. Without much further ado, adios, bye bye, and I hope you enjoyed your weekend. Bye.